Okay, okay. Hello, everyone. Welcome to our seminar. Just one quick announcement, and I think I will start with that because otherwise I will forget. We have an emergency during next seminar, so our speaker is not able to participate and give a talk. So yeah, if you want to give a talk in 2 weeks or by the end of the semester, please let us know. You can email me or Rachel. I think mails are available easily, and you can just Google for us. And yeah, we still have some slots. In 1 month, we already know what we are going to talk. We'll talk about cryo-EM structures of group 2 introns, so it should be a lot of fun and very structural work. But today we go deep into deep learning. So today our speaker is Chaitanya Joshi, and he's a PhD student at the Department of Computer Science, University of Cambridge in England, and he's supervised by Professor Pietro Leo. I hope I pronounce it correctly. So please, let me help. And his research explores intersection of geometric deep learning and graph neural networks for application in biomolecule modeling and design. And previously, he did his undergrad in computer science from Nanyang Technology University and worked as a research engineer at the A-Star in Singapore. So yeah, I'm super excited. Very hot buzzwords in the bio. So yeah, I'm looking forward to your talk, Chaitanya, and the floor is yours.
Thanks, Martin, for the introduction, and thanks to both of you for hosting this really awesome reading group as well. I think, I'm not an RNA biologist, I'm a computer scientist, and um, through this reading group, I've become really interested in RNA. And before that, these were just some of the books that I was reading around the pandemic that sort of motivated me to explore RNA as I began my PhD later. So many of these RNAs that are really interesting are also structured, right? I really like this talk by Riju Das on structured RNAs and a potential revolution in RNA structure determination. Um, and so, we obviously we care a lot about RNA structure at this special interest group. So I don't need to motivate this too much. But I want to talk about what's happening in the sort of protein design world and how deep learning is really starting to work for protein design from a structural perspective with very prominent papers and even corporate investment into this topic. And the question I want to ask in this talk and sort of through some of my work is, well, what about RNA? Can we bring similar advances to designing RNA from a structural perspective? And so the title of this talk is gRNAde, which stands for Geometric RNA Design. It's sort of still in preprint, which is not really up to date. The code base is more up to date. And there's also going to be a book chapter on sort of how to use this technique soon as well. So before I dive in and really describe some of the background and methodology, if you stop listening after this executive summary slide, I hope you'd still take something away. So the idea with gRNAde is essentially, you take as input one or more RNA backbone structures, you strip off their nucleotide identities, and you can then generate sequences that the model thinks are likely to fold back into that given backbone structure, essentially the inverse folding problem. And the tool is essentially this black box within which it consists of a graph neural network, which is specialized for handling the 3D structure of these RNA backbones, as well as sort of incorporating some design decisions on handling the multi-state nature of RNA, followed by a sequence decoder, which essentially predicts the identities of each of your nucleotides. In terms of the main computational results, what we found is that when we compared apples to apples with Rosetta, sort of classical physics-based tool for doing the same task of fixed backbone redesign, we were able to improve sequence recovery and I think also hugely gain in terms of speed or in terms of inference latency. And lastly, we want to build essentially a protein MPNN analog for RNA. And all the code is open source along with a sort of 101 tutorial to this tool and to this topic as well. So that was the summary, and I'll dive in now, and I think I'll start with a term that I don't really like, but generative AI, which is sort of starting to work for protein design. I'll describe a structure-based protein design workflow using deep learning tools. And then I'll talk about RNAs right after that. So essentially how it works is that you start with some template, perhaps from the PDB or from something that from somewhere else. And you'd need to figure out which parts of this template, which chains you want to design. So you'd throw away the identities of the amino acids for those chains that you want to design. And then you'd use this inverse folding tool called protein MPNN to take this backbone and predict the sequence or design sequences that are likely to fold into the same structure. Now, to sort of complete the computational pipeline, you can then forward fold your designs with AlphaFold, and you can then superimpose and verify that your predicted sequences as structures now sort of overlap or match with what you initially started with. And this approach has really begun to work, and there have been sort of a few papers experimentally validating this approach as well. Now, what's also not shown among these line of tools is protein language models, which is a purely sequence-based approach to design. And it's also hugely exciting, but just not covered here. The analogy is to systems like ChatGPT, where these are essentially natural language models. But what we're interested in are these protein language models, or which are conditioned on structure. So you're given some backbone structure, and you're trying to predict the next tokens, the probability of the next tokens amino acids. So you may have, for every sort of step of your decoding or your language, you may have a probability distribution over the 20 possible amino acids. And essentially that gives you these inverse folding sort of tools. The other paradigm that I'm not going to talk so much about today, but is also very exciting, is sort of de novo design, or starting from scratch, wherein you start with some target or some design constraint, and you now have another neural network, another sort of generative model to generate the backbone. So you generate the backbone structure, and then you inverse fold the same backbone as well. And this sort of de novo design pipeline can be seen as a sort of more ambitious version of what I'd previously shown. Now, here the analogy is to tools like DALI, which under the hood are actually using very similar generative modeling approaches as what's working within this RF diffusion tool for protein design. So with that being said, right, what about RNA, right? Like, what is the state of RNA design? And, can we do any deep learning there? Well, for RNA structure modeling and design, as you may know, there's a huge emphasis on secondary structure, right? And perhaps rightly so. I mean, tertiary structure is really hard to determine, and there could be a lot of design problems that we could solve with secondary structure alone. But there could also be certain design problems and tasks which are inherently tertiary and inherently require 3D structure-based tools. Good motivating example are aptamers, which are highly specific binders to biomarkers and can be used as biosensors and so forth. And their whole sort of functionality is based on taking up highly specific 3D structural conformations that will allow them to bind very specifically to their targets. Um, so, so there are relatively fewer tools for 3D design. One of them is called RNA-Make. Now, this is not a deep learning or machine learning based tool as far as I'm aware. RNA-Make essentially uses classical algorithms to assemble these motifs together, flanked by helices along both sides to sort of iteratively build up RNAs. And this is really cool, and it's been shown to work in vitro. Um, Here's an example of how they sort of locked an ATP binding aptamer into its binding position, and showed that you could improve its binding activity. And they have other design scenarios as well. But RNA-Make seems to be very focused on this problem of grafting or essentially scaffolding some motif and building a structure around it. Um, we wanted to think about, maybe using deep learning for other kinds of tools for RNA design, and start trying to look at, what exists within this sort of pipeline for RNAs. And so essentially this talk is about gRNAde, which is an analog of protein MPNN. It does inverse folding for RNA. There's nothing public on sort of the RF diffusion front on sort of designing these backbones or generating these backbones. And I think that when it comes to structure prediction as well as language models, though, I think this is where a lot of people are trying to push the frontier and are working on building better tools for RNAs, or specifically for RNA as well. So this is a wide ecosystem of different types of models and different types of tasks that do need to come together to I guess start doing something for RNA 3D modeling and design with deep learning. And the first question when you want to do any deep learning, of course, is what data exists, right? And the answer is not very much. This is the main challenge of working with RNA 3D data. I've just taken some numbers from two prominent papers, and obviously there's just not that much RNA 3D structure out there, right? Well, in our work, we sort of relied on this repository called RNA Solo, which is a repository of PDB-derived RNA-only structures, which they've extracted from various complexes and, of course, from solo RNAs as well. And the bottom line is that there are today about 15,000 or so RNA structures, which can be grouped into 3,800 equivalence classes. Which to me seems like it's sort of just about enough to try to start training some deep learning models. But this is just my intuition, and other people have sort of told me that this is maybe not enough to cover the structural diversity of RNAs, especially when you look at what some of these prominent models for proteins have been trained on, right? These models have been trained on essentially one order of magnitude more data. So that paints a pretty grim picture, and it sort of makes one think, should we just wait? Like, should we just sort of wait for a few years till computationally assisted cryo-EM gets so good that there are lots more RNAs in the PDB? And I think the answer is not necessarily, because again, if we like take inspiration from the proteins field, right? There have been other successful proposals which have been trained on carefully chosen subsets of data while still being able to match the performance of some of these more expensive tools that are trained on everything from the PDB. So with that being said, right, so we used RNA solo, and I'll show you a few statistics about RNA solo. I'll go through them quickly, but we can come back to them if people are very interested. So when we look at sort of what RFAM families are in there, it's mostly tRNAs and ribosomal RNAs. And there are other things like aptamers and riboswitches in there as well. When we think about the sequence length, then most of the things are shorter than, say, 500 nucleotides. And what's, I guess, really interesting is that RNA do adopt multiple conformations, right? And this is reflected in RNA solo as well. This is often critical for functionality, and perhaps is something we should consider for design as well. So here I'm showing a histogram of sort of the number of structures available per sequence. And there is a large amount of sequences which have more than one structure available. And when you look at those structures, right, often these structures can have quite large RMSDs or quite large difference between the conformations reflected in there as well. So here I'm just showing, for instance, the histogram of average pairwise RMSD among these RNAs which have multiple structures. This is the C4 prime RMSD. And you can see that there is some variety, and there are some RNAs which have quite large differences as well. So with that, so considering all that, right, like considering all those sort of challenges of like lack of data and RNAs existing in multiple states, we set out to build this inverse folding pipeline, which I'll now quickly describe sort of how it works, and importantly, how we evaluated it as well. But maybe at this point, I can also pause and see if anybody has any questions or would like to discuss anything before I sort of move on into describing some of the machine learning models. All right.
I don't see any questions.
No worries. So, okay, I'll start describing this pipeline a little bit. So the first thing you need to do is you need to process your RNAs into something that you can feed into a neural network, right? So essentially, as your input is generally PDB file or PDB files, and you're going to process that into a geometric graph, a graph in 3D space. And essentially, the first step for this is that you look at your RNA backbone atoms and you construct a 3-bead representation. You coarse grain it. So for each nucleotide, you only retain the phosphorus C4 prime and N1 or N9, depending on whether it's a purine or pyrimidine. And you'd compute some geometric features for these 3 beads. You'd compute distances, angles, and torsions along all these pseudo bonds that are left over. And then each nucleotide essentially becomes a node in a 3D graph. So you have a graph wherein each nucleotide is a node, and you obviously connect everything along the sequence. But importantly, you also connect those nucleotides which are spatially close to each other. So you look within some radial neighborhood of each nucleotide, and you connect it to all its neighbors that are spatially close to it as well. And that gives you a sort of 3D graph representation of your RNA backbone. If you have multiple backbones, you just do the same operation for each of them as well. Short aside on why we chose these 3-bead representation, well, I found it really interesting that there was some work saying that these pseudo-torsional descriptors are sufficient to fully capture the diversity of RNA backbone conformations. And I thought that maybe using this sort of reduced representation will allow the model to generalize better, maybe compared to, giving it lots and lots of torsions. Like on its own, there could be, I guess, 7 torsion angles if you use all the atoms in your backbone. Um, I did experiment with this later, but that's what we thought at the beginning. So once you featurized your graphs, right, you can now feed that into a neural network. And essentially what you do is for each of your backbones, you get those graphs, and you're going to feed these backbone graphs into a neural net, which will predict the per node probability over 4 bases. So for every nucleotide, I'm going to predict which base it possibly is. And to do so, essentially, we use graph neural networks to process each backbone graph. We apply several layers of GNNs, after which we get these feature vectors or representations, these sort of latent representations of that backbone graph. And essentially, these representations are supposed to capture something about the geometry of that backbone. And then we use another type of neural network called a deep set to combine or fuse together these backbone representations. So we learn how they're combined within our network. And then finally, these combined representations for each node are then decoded to get a per node probability distribution. So a sort of distribution of probabilities as to which base that nucleotide is. And the decoder essentially decodes autoregressively from the 5' end to the 3' end. And it assigns these probabilities, which can then be sampled from in order to design new sequences. So the model on its own, it just gives you probabilities, and then you can sort of sample from these probabilities to design many, many new sequences. I'll also take a short aside to describe what graph neural networks are, because this is what I'm actually more focused on. So GNNs are essentially neural networks laid along the graph structure of your graph. And what they do is they learn to propagate information along the graph. So for every node, you're learning how to borrow information from its neighbors, and you're essentially building some representation of the local subgraph around each node via this process, which is called message passing. Now, what makes GNNs very special for biomolecules is that you also account for 3D symmetries. You also account for sort of how certain quantities for molecular systems rotate and translate covariantly as you rotate and translate your system, whereas other quantities may not, which may stay invariant. So you need to build that into your models. If you're wondering where to start with these kind of models, we wrote a very long sort of survey on this, which was really written for mortals and starting from the basics. So coming back to gRNAde though.
Chaitanya, I have a short question, maybe too technical, so we can go briefly. So, is
Yeah.
there a common way or like framework to implement this kind of networks, or pretty much you start from scratch in Python? I would assume, or is there some kind of way to define the network and implement that, or use some kind of framework for like
Yeah,
technical?
yeah. I mean, I personally use this framework called PyTorch Geometric, which is based on PyTorch, and it's sort of like an extension of PyTorch to allow you to build graph neural networks. And there's also Deep Graph Library, which does the same, but is also compatible with TensorFlow and so on. So I think by now there are really good frameworks to use these models, and I think also like people in the community are generally sharing their code in an open source way. So there are lots of nice resources and references to use these models in new projects as well.
Mhm. Thank you.
And I think like again, if someone wants to get started, we wrote this really long survey on this. We also released a sort of 101 tutorial for these geometric graph neural networks. So that could be a good place to even learn PyTorch geometric if you're interested in this stuff. Yeah. So what gRNAde does is that you take these backbone graphs and you can then get a few sequences for them, right? So the question really becomes what makes a good design, and how do you prioritize designs in silico? Perhaps you want to send a few of them to a wet lab. How do you know what are good designs, right? And the most simple but most widely used metric in the proteins community, as well as in a few papers on RNAs, is sequence recovery. So with your backbone graphs, you also have this sort of true sequence, right? You don't use it as part of your input, but you do have the true sequence for that backbone. And you have a bunch of sampled sequences. So what you can do is you can compute how accurately these sampled sequences match the true sequence. And you could perhaps take the average over that. So you're essentially trying to see whether you can perfectly recapitulate the true sequence. Of course, there's a caveat to this, that maybe getting perfect recovery is not the aim of good design, because you want to have some amount of diversity compared to the native sequence. But this seems to be like the most common metric that people use. The other thing that we do have is also the true secondary structure, right? So we know which nucleotides are paired and unpaired in the least case. And what we can do is that we can sort of forward fold our designs. So we can use, say, EternaFold or any other secondary structure predictor to predict the secondary structures of our design. And then we can compute a self-consistency score between the true secondary structure and the secondary structure of our designs. And it's essentially trying to measure whether, even if you don't perfectly recover the native sequence identities, can you recover the base pairings? Can you recover the rough structure that your RNA might fold into? And a caveat here is that you obviously inherit all the limitations of whatever secondary structure predictor you use. But ideally you would have liked to do this with tertiary structure, not just secondary structure, but you would have liked to do this kind of self-consistency with tertiary structure. But I think that secondary structure tools for RNA are a lot more reliable, at least at present, compared to tertiary structure tools. So I started by using Eterna. And then the last design metric that one can use, and I'll show is quite interesting, is perplexity. This is the model's sort of guess of the conditional probability of a sequence given a structure. You could understand it a bit similar to PLDDT for AlphaFold, but in this case, it's something really that you analytically get out of your model as your model's confidence for its prediction. So that was gRNAde. That was an overview of this sort of system. And so we set out to do some things with it. What it does is fixed backbone redesign, right? It's a very simple task, I guess. It's like it only scratches the surface of perhaps what real RNA design would encompass. And it's maybe a synthetic setup in some ways, but it's a starting point. What it does as input is you give it a native PDB file, and as output, you can get designed sequences. So we wanted to benchmark this, and to do so, we looked at 14 RNAs of interest, which were identified in this paper by Das, Kara, Nicholas, and Baker from 2010, which was a paper on using Rosetta for RNA structure prediction as well as for inverse folding. So these were 14 high-resolution RNAs from the PDB. And we set out to do an apples-to-apples comparison with gRNAde and Rosetta. And what we found when we compared the sequence recovery was that we were able to see improvements compared to Rosetta. We were able to improve to a median of 54% sequence recovery compared to Rosetta. And perhaps what's maybe more interesting is that we're able to be significantly faster in terms of inference. So, well, once you train a neural network, right, it's really fast. You just run it on a GPU and you just do a forward pass through your neural net to get designs and to get a bunch of samples. So you could, in under 1 second, you can process RNAs with hundreds of nucleotides. Whereas for Rosetta, the same computation may take order of hours. Now, I haven't run it myself, but the Rosetta documentation said that anything greater than 10 nucleotides may take minutes or hours. So I kind of see this as, you know, one of the bigger promises of neural nets is that if you train them on good enough data, then they're extremely fast in terms of inference. And of course, it's important to note here that we did try to evaluate for generalization, and we did try to be as fair as possible. So we excluded these 14 RNAs as well as structurally identical RNAs with a TM score below 0.45 from our training set. So essentially, we were measuring whether gRNAde could generalize to new RNA structures or new folds that it hasn't sort of seen during training. Now, in sort of maybe zooming in into some individual examples, right? Here I was plotting the recovery of gRNAde versus Rosetta on the XY axes. And I'm shading that by the model's perplexity. So lower perplexity means that it's more confident. Perplexity is its own internal measure of confidence. And it roughly means, like, on average, how many nucleotides is it choosing from whenever it's making a prediction. And what we see is that it seems to correlate quite well with recovery as well, which is interesting. And then, you know, when we look at like specific examples, there are a lot of them where gRNAde has a higher recovery than Rosetta. For instance, here's a guanine riboswitch aptamer, which perhaps, you know, has quite a higher sequence recovery because there could be parts which are highly conserved. There are other examples where these models are neck to neck, and you know, here's something from a ribozyme where, you know, both models perform roughly the same. And there are yet examples where Rosetta does do better, although there are fewer. I mean, we look at a very limited subset, but there are fewer. And then there could even be examples where, now I believe maybe there was something in the training data set that looked very similar to this example, and then gRNAde got almost perfect sequence recovery, because generally we wouldn't really want such a high sequence recovery, right? So it's sort of interesting to see this range of recoveries, and we're thinking about how to most fairly compare to Rosetta and to previous design tools as well. But perplexity is still quite interesting and seems to correlate well with recovery. And what we want to do, and you know, this is sort of work in progress so far, is to then use perplexity to rank fitness of mutations. So it's essentially the conditional probability of a sequence given the backbone structure, right? So what we could do is we could take data from studies with 3D structure, as well as fitness measurements for some mutational landscape. And then we could try to measure the correlation of the fitness with the model's perplexity. And there's at least some evidence in the protein world where looking at these kind of measurements has been correlated with more thermally stable mutations. So this is something that we're still sort of working on, but just an idea that I wanted to float as well. So that was for single state design, and that's, you know, that's almost like a comparison to Rosetta and just the most basic task we could do. But I think what I believe is an emerging view in the RNA community is that it's not just structure, but rather conformational ensembles that determine function, and often the propensity of these conformational ensembles, right? Um, and in this example, right, this is an adenine riboswitch. And for this riboswitch, we have both the bound and unbound structures available, as well as an intermediate state. And you know, there is actually a large difference between the bound and unbound state in this case. So if we were using a single state design tool for this sort of design scenario, I mean, maybe not this exact scenario, but you know, if we face similar sort of tasks, it could be quite ambiguous because single state design tools are essentially optimized for predicting the best sequence given one structure. Whereas we wanted to think if we can explicitly train our model to predict the best sequence given multiple structures. So to explicitly build that idea into our model. And that's what sort of inspired some of the sort of ideas of building graph neural networks that can take as input multiple graphs. So then we, you know, setting out to benchmark this is a bit difficult though, and sort of trying to create a challenging test set of structurally flexible RNAs is what I'm going to quickly describe here. I may not have done a great job at it because I don't have that much of a background in the biology. But here's what I tried to do, right? So I clustered RNA solo with US align, and then I ordered these clusters based on median intra-sequence RMSD among the available structures in that cluster. So for each cluster, you'd have some RNAs, and each RNA could have multiple structures. So then I computed the RMSD among these multiple structures, and then I looked at the median within the clusters. And so then I can make my train validation test splits progressively harder or progressively more flexible. So the top 100 samples with the highest RMSD would go into my test set. The next 100 would go into validation, and everything else would be kept in training. I'd also keep RNAs larger than 1,000 nucleotides in my training set. And if anything was not clustered, I'd also send it to my training set. So that left me with around 4,000 RNAs to train and 100 RNAs each in my validation and test sets. The idea behind this procedure was to essentially have validation and test sets where for a given RNA, you can have multiple structures. So the number of structures varies, and such that you have reasonably high RMSD between those structures for your val and test samples, such that you're explicitly evaluating whether your model can recover on these so-called more flexible RNAs or these multi-state RNAs. I think a lot more work can be done into obviously defining what is a state, like what is a conformational state or what is a functional state. But I did this, I think, very naive sort of approach to this. So then when we sort of trained models and evaluated them, we essentially trained gRNAde with 1 state, 3 states, or 5 states, so up to 3 states or up to 5 states as input. What we find is it gives very small performance improvements overall. Like all these sort of models, single state or multi-state, they don't have very large performance difference among them. Obviously, the native sequence recoveries are a bit lower now because these are perhaps more challenging RNAs. And similarly, when we look at these self-consistency scores based on these secondary structure metrics as well, again, there's not much difference. I think generally the multi-state models have like are slightly better, but not by a lot. And then the other thing to note is that when we look at these self-consistency scores, right, these are actually Matthew's correlation coefficient between the true and predicted secondary structure. So these can actually take on negative values as well. But what was promising is that at least none of the designs take up negative Matthew's correlation coefficient scores, which means that at least like they all lead to somewhat reasonable secondary structures. So these results are sort of saying that there is room for improvement in how we're designing these multi-state models as well as how we design their evaluation. I think, then what we're showing here though is at a per sample level, but we can also evaluate recovery at a per nucleotide level as well. So we can, zoom in and sort of look at whether the model is recovering particularly challenging nucleotides in a good way. So something very simple that we can do is we can characterize nucleotides in terms of their solvent accessible surface area, as whether they'd lie on the core or surface, maybe. And what this plot shows on the y-axis is the native sequence recovery, and on the x-axis is the SASA value for the nucleotide. I've binned it into 10 bins or so. And then the red curve is the proportion of nucleotides with that particular SASA value. And then the models are in the other colors. And what we saw is that if you look at some of the bins where the SASA values are quite high, so maybe these are nucleotides on the surface, then we saw that the multi-state models are slightly better than the single-state model. And maybe that's where their performance boost is coming from. It might be coming from these nucleotides, which are maybe more flexible or lying on the surface. We tried to look at also paired versus unpaired nucleotides. So now on the x-axis, we plotted the pairing probability. So, if it's 0, then something's highly unlikely to be paired, whereas if it's 1, then it's always paired. And if it's somewhere in the middle, then it's maybe more ambiguous. And here we saw that, again, multi-state models are better recovering paired positions, but perhaps more interestingly, also positions that are mostly paired, but not always paired. So there's some sort of change in secondary structure. And we could see that multi-state models help over there as well. We also tried to, again, do the same thing, but with RMSD now, like with nucleotides, which are varying in position, and we could see similar trends. So the idea is essentially that these multi-state models might be giving you a slight advantage when it comes to some of these more challenging situations or more structurally variable regions of your RNA. With that, I'd like to conclude and sort of ponder about things that we're actively thinking about. The main thing is, this is all in silico, and we'd like to validate this in the wet lab. And I think that's where these sort of tools will really be sort of pushed to their limit, and new ideas will come from. I think some of the questions that then arise are, how do you choose the number of states, and what is your exact design scenario essentially? For instance, if your design scenario is similar to the RNA make situation where you're locking something into a bound state, then maybe that's just a single state design problem. And then secondly, how do you prioritize among design sequences, right? So perplexity and so forth, you can use that to rank sequences, but perhaps in real world settings, right? These design tools are often being used in conjunction with yet another machine learning model to select among designs. So, almost a fitness predictor, which might be based on experiments that your team conducts, and then you can hopefully train another ML model to select among designs. So I think really taking these tools to the wet lab might lead to more innovations. I think there's limitations in terms of the methodology, though. So for instance, we should in the future try to support multiple interacting chains or account for interactions with ligands. I mean, currently we just sort of extract these RNAs out of protein RNA complexes mostly, but it would sure be nice to account for that sort of protein partner when you're designing the RNA. And essentially, try to improve how we benchmark multi-state design as well. We have released all the code and checkpoints on GitHub, along with a tutorial on how to use this tool, as well as a book chapter forthcoming as well. I'd like to thank my lab mates and my supervisor at Cambridge, as well as some senior people who I talked to and sort of who inspired me to work on this stuff. Roger Fu, Phil Holliger, Alex Borodovka, and Janusz Bujnicki. And thank you for listening as well. And I'm always happy to take questions here as well as offline.
Okay. So I work with Janusz, so it's Bujnicki, so everyone can learn. I think Riju wants to, or just clapping. Riju wants to
Yeah,
say
can I that was a really nice talk. I was curious about the the grenade model. In principle, it can learn principles of to avoid incorrect secondary structures because it ends up passing messages, I think, throughout the entire polymer. I was kind of curious, have you looked at sequence recovery for single structure design with and without that eternal fold filter? Like, does it really matter?
So we aren't using Eternafold to filter things though currently, or perhaps you could clarify, like, do you
Oh, I
mean
see.
like,
So you're using it as a score later. I see.
yeah.
If you have a cut where you say I want a self-consistency score above a half or something, then does that change the sequence recovery? Does it make it better or worse? Is there any signal?
I haven't looked at exactly that, but that would be really interesting. I think having this kind of cut, um, you know, one thing anecdotally that I have been seeing is that sometimes the Eternafold self-consistency score can be anti-correlated with recovery. So sometimes having really high recovery could mean having quite a low self-consistency score, which is strange, but it might mean that there are parts of the sequence that you recover perfectly, but then there are these important parts that you missed out on that led to Eternafold predicting a different secondary structure. And
I think the issue is you may be seeing is that maybe the best way to stabilize a helix is to put in a bunch of GC pairs.
Mhm.
But that also leads to the prospect of having a lot of mismatches between a G and a C that's in the wrong part. That's the wrong pairing. And so I wonder if that's what's giving rise to the anti-correlation. It's pretty fascinating.
and I
You'll find
mean,
out in the wet lab.
this is not just something that I'm observing though, so in the protein design world, sort of with protein MPNN and so on, right? They have something very similar, right? Like, I mean, they have this idea of forward folding, or essentially designability, right? Like where you get your predicted sequences and then you alpha fold them, and then you're computing a score that they call designability score between the prediction and the input backbone. Um, and what people have seen is that recovery, so native sequence recovery may be less correlated with success in the wet lab compared to this designability score. So some papers have started arguing that we should prioritize this sort of self-consistency designability score compared to native sequence recovery, um, which I guess is you can only see this in the wet lab, right? Like this seems like the limit of computationally evaluating these sort of design tools, right?
Thanks.
Okay, Janusz Wojnicki.
Thanks. Great talk. Chaitanya, we have a great discussion at the meeting at the MDL. Now I have actually one broad question.
Mm-hmm.
Of course, recovery of sequences from known templates, known faults,
Mm-hmm.
is one task. But I think the biggest splash that David Baker's Rosetta did in protein design was when they were able to design a protein that has, I mean, almost a completely new fault. It was not like completely new, but it was different from other structures. So what do you think about, first, the ability of your method to design RNA 3D structures that are like very different from the existing ones, and not only in the architecture of major secondary structure elements, but also in the new arrangement of non-canonical interactions? This is one question. And second is about the difficulty. How difficult? What is your what do you think? What would be more difficult, to just redesign the existing faults, or design like something completely new? I understand that the difficulty comes in actually who would come up with something really very weird, intricate 3D structure. But I mean, leaving this aside, to extent your method would deal better with known backbones or like totally new backbones?
So, you know, as far as it comes to like what I talked about here, right? This grenade model, it only does inverse folding, so it can't it can't design new structures or new backbones. It can only sort of inverse fold them. But you're right, like the most exciting splash in protein design came from here, right? Where you had this neural net that could now generate new types of backbones. And they even showed that many of the backbones that they were generating were sufficiently different from what's already in the PDB, right? I mean, my intuition for how those models work is that they essentially mix and match substructures from the PDB. Kind of like the RNA make algorithm does as well. So of course, I've written that there's nothing public in this realm using deep learning yet, but I'm sort of working on this and really interested in building these sort of things. Perhaps based on ideas like what RNA make did, right? Essentially trying to think about fragments and so on. I think it's a really hard task, right? Like, I think there's a lot of intricacy to RNA 3D structure, and I mean, I don't claim to fully understand it at all, but there's there's just so many small little things that RNA biologists sort of look at in their talks that I don't know if we have enough data to fully learn all the intricacies of sort of how these tertiary structural motifs come together. Like even if we think about like the last talk in this CASP SIG, there were all these tertiary contact motifs that Joseph Yesselman was talking about, and I don't know. I think that would be really difficult for a neural network to learn precisely. Which is why I kind of like the fragment-based approach. Like if you can maybe use fragments, it might be more tractable. Whereas in the proteins world, right, they had perhaps so much data that they could just learn all these substructures from the data itself. But I mean, at this point, I'm just hypothesizing. I think that it's a hard problem, and I think something I'm very interested to try to work on as well.
If I just may add to this, I think the fragments could be considered not only in the sort of, linearly connected parts of the backbone, but different fragments coming together. So I think fragments like, loop receptor interactions are pretty well understood. So I think as this is actually something that people have worked on for a very long time, and it's pretty well understood. So I mean, if we can recapitulate this in the design process, I think this could lead to better design of these long-range interactions. So anyway, good luck with your project.
Yeah, no, thanks for the discussions.
We have a few questions in the chat. So first one is from Shrikan. Nice and informative talk. We all agree that was great. Thank you very much. Why sequence retrieval from multiple structures? Did you account modified residues present on RNA PDB structure? Yeah, that's kind of always.
No, I didn't, and I think that that's something that I should do, but at the moment I sort of just threw away information on modified nucleotides. But as we know, they're really, really important, right? How could one do that? I mean, how does one incorporate that? Perhaps it could just be added as new vocabulary elements. But I think what then happens is that there's so few of them, right? That you have this very big data imbalance sort of problem. So, I mean, I don't know what's the best way to incorporate modified nucleotides as well.
Yeah. And the last one is, may I ask why you chose deep set specifically to combine embeddings from different backbones?
Yeah. So I'm not sure if your background's in the sort of machine learning or the RNA biology, but if you're working on machine learning, right, then this deep sets thing that I did, you're right, it's the most simple thing we can do. So deep sets are a type of neural network that take as input a set of anything, like a set of inputs, and then output a single representation or a single prediction for that set. While in what and the main property that deep sets have is that they're unordered, so they don't consider the order of your input set. And that's what we wanted here, right? Like we wanted an architectural element that would not consider the order of the backbone. So you could shuffle them, but your model should not sort of give you a different output for shuffled ordering of your backbones. So the deep sets model was the simplest thing we could do. But if you're really interested in sort of the machine learning of on set stuff, which I really am, then there's another architecture called deep symmetric sets, which are generalization of deep sets to other symmetries, which we have in this case, right? Because we process the inputs to our deep set with a GNN, which also sort of needs to deal with 3D symmetries. So deep symmetric sets are sort of more expressive version of deep sets. But when I tried it, I think that they overfit really well to the training set, but we're not generalizing well. And I think that's the central problem of sort of doing ML in most real world problems, right? That like the more expressive thing that theory suggests you should do often overfits your training data, whereas maybe like simpler methods, which are better regularized, can generalize better. So that's why I currently went with deep sets.
So maybe the last one from me is I don't know, have you checked the recent paper of Atomic AI about Atom 1? So what they are using is this, I think, I don't know, it's fundamental model or like probe networks. But what they claim that even for very, very limited set, this network is able to find like fundamentals of like physics and chemistry. So their argument is that this kind of architectures they are
Yeah.
able to generalize very, very well, even on limited sets. Do you think that this is going to answer like all of like some of the questions that we have about very limited set in terms of like shapes, in terms of like, as we discussed before, of modified residues, or what's your take on these networks?
I really like the vision of that paper and generally of that line of work, right? Like it's essentially like the scaling hypothesis of deep learning is that if you can find a task where you can scale the data and you can scale the model parameters, and the task is sufficiently hard that you can learn general enough properties about your data, then you have this foundation model which you can then fine tune and adapt to many downstream tasks in a more data efficient manner, right? You could have fewer data for those other tasks. And I guess like I mean, the atomic AI paper in particular, just I don't think it's very like it's completely scientific in that they don't tell us how many RNAs they pre-trained on and what the data source was, or it doesn't go into all the details that I would want it to go into the details of. But I guess that's understandable because this is the times we are living in AI research. But I really like that idea of using these sort of these structure probing experiment data to train a language model, and then use its embeddings to do other RNA tasks, because structure probing data seems like something that we can scale. So I participated in this Ribonanza contest as well, and I didn't do too well, but it was still a fun way to learn about this area. And like just think about what could be the potential of this kind of structure probing, cheap but very large scale data. Um, I think that, with what's already out there with Ribonanza, and perhaps, Ribonanza 2, that's promised to be much larger, a lot of the rest of us could explore ideas similar to atomic AI as well. Because, they're a company, so they haven't really told us how they did what they did, right? So I think like I'm still skeptical till I or others can try it out in an open source way, but I really like the idea.
Yeah, we should get Ritu back and talk more about the results of Ribonanza.
Yeah.
But yeah, soon to be discussed. Okay, I don't see more questions. I think it was great, very exciting times to be alive. Enjoy all this progress in the field. And as before, we are looking for speakers. So let me know, let Rachel know. I hope Riju will talk more about Ribonanza, because it seems now it's a very, very interesting, I think topic going on in the community. And
thank you, Martin and Rachel, for running this show. This is an exciting series of seminars. Thank you very much. You